Hello, everyone. Welcome to today's live broadcast, Validation and Commercialization of Liquid Biopsy in a CLIA Lab. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Mr. Michael Nall. Michael Nall has over 25 years of healthcare sales and marketing experience, most recently serving at CLIA Diagnostics Services Incorporated, in positions of increasing responsibility from 2002 through August 2013, with his last position being General Manager, North American Sales and Marketing. While at Clariant, Mr. Nall was also responsible for leading the team assimilating Clariant into GE Healthcare after Clariant was acquired in 2010. From 1988 until joining Clariant, Mr. Nall served in the diagnostic and medical device industries in various commercial leadership roles for companies, including Impath, American Cyanamid, McKay Surgical, Strato Medical, Horizon Medical Products, and Columbia Vital Systems. Mr. Nall received a Bachelor of Science degree in Business Administration from Central Missouri State University, now known as the University of Central Missouri. I will now turn it over to Mr. Nall for his presentation. Mr. Nall?
Darby. Folks, I'm excited to be here and share the story of Biocept, where we're delivering personalized medicine through a liquid biopsy. And before I get started, I always take a couple minutes to explain to folks what do we mean by this liquid biopsy concept? Because if you Google it, you're going to get a lot of hits. Well, at Biocept, we help patients who have been diagnosed with cancer make sure that they get the most appropriate treatment by their physicians. So their physicians send us a simple blood draw, and we can get personalized medicine information from that blood draw that they use to make treatment decisions for their patients. So a little bit about our company at Biocept. Well, we have 18 patents on the testing that we do. We're focused on two aspects to the tube of blood that comes in. There's circulating tumor cells, which are intact cells that are shed from solid tumors into the bloodstream, provide a source that we do specific genomic and proteomic analysis, as well as cell-free circulating tumor DNA. So all the cells in your body are breaking down constantly, and when they do so, they release nucleic acid into your bloodstream. Cancer cells are the same way. So we can isolate those rare cancer DNA fragments out of the blood and analyze those on the genomic basis. So really two areas we look at, both with patented technology, intact cells called circulating tumor cells, as well as cell-free circulating tumor DNA. We provide this information that physicians use to make treatment decisions for their patients to make sure they get on the most appropriate care, get the most appropriate care that they can. We do this testing in our own laboratory. It's called a CLIA lab, which stands for Clinical Lab Improvement Act, and it's CAP accredited as well, the College of American Pathologists. So high quality results that come out of our lab as they inspect us every other year to make sure we meet these stringent quality standards. Now our lab's located right here in San Diego. We get specimens FedExed in from all over the world, and we do the testing right here. But in the future, we want to go through the FDA so we can then market our testing to be done in hospital labs and labs all around the world. So that's in the future at Biocept. Now we've grown very fast because liquid biopsy is addressing a true unmet medical need for patients, and we'll talk more about that in a bit. But our volumes have grown as our revenues have grown as well. And we've done that with a lot of collaborations. We aren't doing it alone. We have world-class institutions that we collaborate with various different studies, from University of California, San Diego, Morse Cancer Center right here, to Georgetown, MD Anderson, Dana-Farber, many others that we either have active or have had collaborations and research studies with. So this is a very large market, and I'm on slide number 2 here. This is a very large market that's estimated to be between $10 and $20 billion by the big banks. Goldman Sachs, UBS, Bank of America, JP Morgan, they all have analyst reports just in liquid biopsy. And they say it's going to be very big. And that's because you're taking testing that's normally done one time only for a patient, maybe two times, with a biopsy procedure, surgical procedure, and we can deliver that same information with a simple blood draw. And the key is the blood draw can be done over and over and over to monitor the patient. So that's one of the biggest advantages of using blood versus a surgical biopsy. A physician isn't going to turn their patients into a pincushion and biopsy them over and over. Versus a blood test, you can test that patient every time they come to their physician. Now, we focus on the most clinically actionable information. So in other words, we say it's the biomarkers that we have. And biomarkers are the genomic signatures that you look for to see what therapies a patient can possibly qualify for. So examples are HER2, and you could qualify for Herceptin from Genentech. EGFR could qualify you, EGFR mutation could qualify you for Tarceva from Genentech. ALK translocation could qualify you for Xalkori from Pfizer, et cetera. So there's a whole bunch of these different, what we call targeted therapies, and you need to know this personalized biomarker information. This is what we test the CTCs, the intact circulating tumor cells, as well as the cell-free circulating tumor DNA for. Now, we do this in a very specific and sensitive format with our proprietary and patented technologies. So industry-leading sensitivity and specificity in the testing we provide. Of course, this is leading to high growth. Oncologists need this information, and they like the way we deliver it, focused on these markers that they know what to do with. Now, because we are able to develop these assays and perform them in our own lab, we can really drive costs down and actually someday have a margin as we get to scale at the business that we do right here in the laboratory. This creates a lot of opportunities for value-creating partnerships both here in the U.S. and outside the U.S. Many of the patents we have are in other countries such as China, Korea, the EU, even Australia. So a lot of opportunities to create more and more partnerships outside the U.S. And as we said earlier, we want to eventually move this to a kit format where hospitals all around the world can do it. So just to make it clear, what we're able to do at Biocept is take what normally requires a surgical tissue biopsy to get the personalized molecular information that's needed for treatment decisions, all from a simple blood draw. That's what you see illustrated here, as well as a sample of the report that goes back to the physician who ordered our test, and they use that to make treatment decisions for their patients. So why would someone use a liquid biopsy at Biocept, which is a simple blood test? Well, first of all, many patients are too sick, so we're truly addressing an unmet medical need for those patients. Those patients could be too sick to even undergo a surgical biopsy. So in that case, a liquid biopsy is able to provide the information that's simply not available if the biopsy can't be performed. So that's a true unmet medical need we're addressing. In addition, even if a biopsy is performed today, especially in lung cancer, many times it's done with what's called a fine needle aspirate. In other words, a very small needle biopsy that's then used for not just the molecular testing, but also to make the diagnosis. So very often, there isn't enough tissue available in lung cancer to actually give the information. So that's also true when other cancers become metastatic, or spread. There's very often just not enough tissue to get the information that they need to make the treatment decision. We address that unmet medical need, providing that information from a simple blood test. If a patient has metastatic disease to the bone, it's very difficult to get a biopsy, and if so, very often can't be counted on because you can't trust a negative result due to the processing of that bone specimen to do the testing. So we can help there as well. And finally, there's a concept of heterogeneity, both intratumor heterogeneity as well as intertumor discordance. And really, those just mean differences between either the biopsy itself, as we look at different parts of the biopsy, or within different sites of that metastatic disease. For instance, if there's bone and brain mets, you might have different genomic signatures. So with a blood sample, we're getting cells and DNA shed from all over that tumor. And therefore, we account for that heterogeneity with what we see in the blood. And the easiest way to understand that is to picture a loaf of raisin bread. And so if you had a loaf of raisin bread and you put a needle in, you might hit a raisin, you might not. So that's the same with the tumor. There's a lot of heterogeneity within a tumor, so as the physician does the biopsy, they could be missing important drivers that would be in a different part of the tumor that wasn't available to them. So with the blood test, we help to address for that. But one of the most important advantages that a liquid biopsy has over a tissue biopsy is cost saving. You know, even with our testing, a liquid biopsy costs about $1,300, and that's on average with what Medicare or the private payers would pay us. A tissue biopsy in America can cost up to $16,000 in a lung biopsy, and that's when everything goes right. It's not just the surgery charges, it's the hospital charges, pharmacy, pathology, radiology. If you've ever had a procedure, you know all that all adds up. With a liquid biopsy, we have the opportunity to save a huge amount of money for the healthcare system, which is so important in this day of the age of healthcare cost constraints. So this is a big potential market we know of. It's based on both profiling and monitoring for liquid biopsy, and we all know folks who are afflicted with cancer. And as we know, there's more and more diagnosed every year as the baby boomers come into their later stage of life. So today at Biocept, and I'm on slide 6, by the way, we're looking at several aspects to this large market. First one is profiling. And that's where most of the specimens that we get in today are in the profiling area. And that's where we help get an answer for patients when they're newly diagnosed or when their tumor recurs. We also are starting to get a lot more business from monitoring for signs of progression of disease or response to therapy, we hope. And they can do that because the tumor cells and the tumor DNA in the blood are tied to the tumor themselves. So if the tumor themselves is going away, therefore you would expect that there's not tumor cells and tumor DNA in the blood that are persistent. So that's an important aspect as well. Finally, companion diagnostics as well is something that we're doing at Biocept. We're involved with 2 pharma trials today where we developed assays for early stage drugs as they start to explore the impact of their treatment. If the drugs are successful, that puts us in a good position with the assays we've developed for them and a potential licensing opportunity as they move their drugs through the regulatory path. Finally, there's screening. And today we aren't in screening at Biocept, but that's an opportunity in the future. A big opportunity for screening, but a lot more data needs to be gathered with screening as we find out what it would mean if we found small amounts of DNA or circulating tumor cells in an otherwise asymptomatic patient. So in other words, if you or I would have an annual test to find out if we might have cancer in our blood. Sounds like common sense, but in reality, at this point as an industry, we don't know what to do with that if we found it, because your immune system might take care of it, laboratory surgery, full body scans. So screening is in the future for everybody in liquid biopsy. But today we're focused on profiling, monitoring, and companion diagnostics. So at Biocept, we're the premier commercial platform in liquid biopsy, and that's because we've got this patent protection around what we do. Very high performance with the validations, both in sensitivity and specificity of our testing. Very strong growth delivered by our world-class commercial team. Getting paid more and more every day as we gain more and more contracts. Recently just gained a contract with one of the Blues plans. Very happy about that. Additionally, an opportunity to drive costs down out of the systems and keep our costs with what the payers are willing to pay for this testing. And finally, and someday in the future, we'll be looking at this as kits that we can then market to hospitals and labs all around the world and let them do the same testing we do in our blood today. So when you see the next slide, you can see it looks kind of like a tree, but it's actually a great diagram that came out of nature that shows your circulatory system. And so I love this diagram when we do these talks because it helps to indicate what we're actually analyzing, you know, in a cartoon fashion. So you can see over on the left side of the screen, or the left side of the image, there's a mass, and that's the blue part. That's shedding DNA, as you can see there, cell-free DNA, as well as towards the top, an intact cell, a circulating tumor cell. These are both important to capture and analyze because they both have unique things you can get out of them. So with the circulating tumor cells, we can get protein expression, we can get DNA amplification. These are all tests that require the context of an intact cell. So that's important. From the same tube of blood, we get both intact cells, the circulating tumor cells, as well as that cell-free DNA, which you can get out of the plasma. And that's what you see down below on the other. And you can get mutations and copy number out of that. All important information for a patient and all needed by that physician to make a treatment decision. So when a tube of blood comes into Biocept, we spin it down into its components. You have the plasma on top of that spun down blood, and we do the mutations from there. So that's where we use our fragments, the circulating tumor DNA. In the middle of the components, you have what's called the buffy coat, and this is where the nucleated cells land. because when you go through centrifugation with a blood sample, the light part of the blood goes to the top and the heavier part, like the red cells with the iron, goes to the bottom. In the middle, the nucleated cells. This is where the circulating tumor cells are. Primarily, it's white blood cells, the normal cells. There may be 10 million white blood cells and yet only 10 to 100 tumor cells. So how do we get the 10 to 100 tumor cells out? That's where our proprietary and patented technology comes in. So first, though, let's take a look at what we do with plasma. And by the way, when I mentioned the patents go all the way from the vacutainer tube we use to collect the blood in. It's got our media inside, and that media keeps the specimen viable for up to 96 hours for circulating tumor cells, up to 8 days for circulating tumor DNA. So that allows us to get specimens not just from here in the U.S., but from all over the world through customs. So we can then do the testing in our lab here in San Diego. You see next is our microfluidic channel, and that's the size of a microscope slide. It's clear just like a slide. That purplish color you see inside is dye, so you can see the fluid path, but that's how we capture the intact cells. We also then stain them and identify them so we can know what they are once they're in there, and some of that is patented as well. And finally, our CellFree process has patents pending. Now, with the CellFree process, it works in a very elegant way to totally suppress the normal DNA. So an easy way to understand that when you're looking at DNA fragments in your blood is that all the cells in your body are constantly growing and dividing and dying and breaking down. When they do, they release all the DNA and nucleic acid into your bloodstream. Now, when we isolate that DNA, there's vastly more normal DNA called wild type. That's what you see the WT there. Wild type is your normal DNA. It has a lot more of that than it does with the cancer mutated DNA that we're looking for. So in other words, we have a classic needle in the haystack scenario. So the way that our platform works, it's a chemistry that actually suppresses the normal DNA and only allows amplification, in other words, detection of the mutations that we're looking for in a certain region of the DNA. So in other words, if you picture it like a switch, the switch is off on the normal or wild type DNA, and the switch opens up and allows amplification on the mutations that we identify. So very high sensitivity, very high specificity, greater than 99% and 95% sensitivity, 99% specificity. And in fact, that's at 1 in 10,000. So in other words, one mutation DNA in 10,000 normal. So very high sensitivity in our assay. When it comes to the intact cells, the circulating tumor cells, it works in another very sensitive and specific way. So once again, the plasma in this diagram has gone off to molecular lab, and that's where we're doing the mutations through the process that I just described. Now with our secondary process for the intact cell, we take that buffy coat and we mix it with an antibody cocktail mixture. Those antibodies latch on to the tumor cells, and those antibodies then are run through our microfluidic channel. We can then isolate the cancer cells that have the antibodies on them within the channel, and then the normal cells, in other words, the white blood cells that we're not interested in analyzing because they're not tied to the tumor, go on through. So therefore, we're left within the channel with an enriched population of tumor cells. So from there, we stain them just like you would with the tissue biopsy that would be done. And you can see examples of the stains that we would use. There's a lot of different stains we can use on those tumor cells. Here's two. One would be for protein, and you can see that's the green cell that you see with the protein expression on the outside of the cell or the membrane. Finally, the next one is the FISH testing we do, and you can see this one happens to be for HER2, and it shows that it is a cancer cell and it's amplified. That patient where that actual cell came from would be eligible for Herceptin therapy. So two different ways we can analyze them, both for protein as well as for genes, all in the channel. Now we can also release the cells if we want to do further analysis on those by advanced testing called next-gen sequencing. Now when I talk about the industry-leading assay performance, there's really two ways to look at that. And we always make sure that we explain this to folks. Because in our industry, there's two different areas. There's analytic validation, which is a controlled validation that you do with a known challenge. So in other words, you know what went in, so you know how good you are at detecting it coming out the other side. So in our case, we take tumor cells or tumor DNA and we spike it into normal donor blood. In other words, blood that we get from people that don't have cancer. So therefore, we know how many tumor cells went in. Therefore, we know our efficiency, our sensitivity, and our specificity at capturing those tumor cells. The same with tumor DNA. You can take what's called cell lines and put them in that blood, and then you know how well you are capturing. And you can see there we've done thousands of different challenges in our analytic validation and have very high sensitivity and very high specificity. But what most people ask about when they ask about sensitivity and specificity is how do you compare to the standard of care, which is the tissue biopsy? So that's a lot more difficult of a challenge. And that's because the tissue biopsy from a certain patient whose blood we would get, and this would be a cancer patient, could have been gotten months or even a year beforehand, before the blood comes to us. So therefore, as the patient's treated, it becomes their molecular profile, the way we're comparing blood, becomes different due to response to therapy and evolution of disease. So even with that, we have very high concordance with the tissue biopsy at 87%, and that's the oncologists feel very confident in ordering our test because of that when we explain it that way. So generally, though, the closer we get to having the blood and the tissue biopsy at the same time, you'll see the higher the concordance. In fact, we have one study that was just presented at the European Society for Medical Oncology that we did with AstraZeneca and with the NIH in Mexico, where we showed 90% concordance in lung cancer with the testing that we did. So the closer we get to the surgical biopsy and the blood drawn at the same time, the higher you'll see the concordance in general. And then when we see the further apart, you'll see differences. Either way, still very high concordance rates with our testing in tissue. So there are other companies that are also out doing liquid biopsy, and actually we all kind of support each other as we all have kind of our own area that we're playing in. With us, since we're focused on both intact cells and cell-free DNA, that's a truly differentiator. We're also focused on the biomarkers that matter. Most of the other folks that you see there are focused on very large panels where they're doing a lot of different mutations, and those are really appropriate for patients where they don't know what else to do or they're trying to get a patient in a clinical trial. Our testing is done just like they'd use the surgical tissue biopsy to get the biomarkers that would be the kind of the standard treatment for them. So that's that. In other words, and our testing can also be ordered in an a la carte format, so physicians can order our tests for the biomarkers they're most interested in and not have to only get a whole panel. So we have assays that fit a patient. Don't try to make our one assay fit all patients. So that's kind of how we differentiate between the others. In addition, there's 13 biomarkers that we validated in what we call our CLIA lab. We can do more and more biomarkers than that for pharma and research projects, but today there's 13 that are available. You see those in the red, and you see some of the other markers we're going to be launching throughout 2017 in our pipeline. And one of the key things is an oncologist, as I mentioned earlier, can order one biomarker, or they can order multiple biomarkers from this menu. So when they do order a biomarker, I always take a minute to explain how we get paid. And the way we get paid is that each one of these biomarkers has their own reimbursement code. So in other words, there's a CPT code for EGFR, there's a CPT code for KRAS, et cetera. So it ranges from about $300 to $400 for each biomarker, up to, if they order all of the biomarkers in lung, it could be $2,500 at Medicare rates. So it just varies on what they order as to what the reimbursement would be. But on average, it's about $1,200 per patient sample at Medicare rates for those cases that come through our door. As I mentioned, we do have a lot of partnerships out there. We currently are involved in about 24 research studies with various folks. Many really luminary institutions recently announced a project we're doing in breast cancer with Columbia, which was key. So we are always presenting at different medical meetings and symposia. We have a lot of great scientific and clinical advisors, very proud about those folks that we've been able to get recruited over to help our company. As well as, so also doing something that was new to me when I came to Biocept, and really an important aspect, I think, for our marketing plan. And that's really working closely with patient advocacy groups. And if you asked me a few years ago what I thought the role of patient advocacy would be, I would have said, well, it's primarily to support the patient. And that, of course, is a huge role they play, and very important role. But that's not the only role. In fact, they're very involved today with diagnostic companies, with pharma companies, making sure patients are getting the most appropriate treatment, finding patients for clinical trials, funding studies. So we're very involved with various patient advocacy groups, from the Lung Cancer Foundation of America to breast cancer groups to the Dario Lung Cancer Foundation. So those are an important part of our strategy, so we can make sure our testing becomes aware to more and more patients, and most importantly, their physicians. So in addition, there's a lot of data that we've gotten out on our testing, both in publications as well as abstracts and posters. As I said, we have so many different studies, and a lot of the data is now coming out that most of the big meetings will have either an abstract, poster, or a presentation tied into that meeting. I mentioned the most recent one we had, which was with the European Society of Medical Oncology, showed 90% correlation. In addition, we showed 100% analytic sensitivity and specificity with our PD-L1 assay that we presented at World Lung. And PD-L1, of course, a very important biomarker today, help to indicate whether someone will qualify for immunotherapy, such as that from Merck with Keytruda. So I mentioned before about some of the differences sometimes you can see between tissue and blood, and that's important to understand. You can see that right here in a real-world case study. And this is with one of our collaborators, Dr. Bazanova, at University of California, San Diego Morse Cancer Center. And Dr. Bazanova is a fantastic oncologist. She's actually on the NCCN Guidelines Committee for Lung. And she was helping us with our validation with our CellFree assay for a certain biomarker called T790M, which is part of the EGFR gene. And it indicates when you become resistant to therapies such as Tarceva. And she was sending tissue biopsies to another large reference lab. At the same time, she was sending blood. Well, it was missed with the tissue biopsy, and I believe in the large reference lab, they have great testing. I think it was probably due to that heterogeneity. If you remember the raisin bread analogy, where you might miss something just with a single biopsy, we picked it up with the blood, and so therefore she did something you would normally not do, except if you're in a teaching hospital like Dr. Bazanova is, she rebiopsied the patient the second time. And sure enough, when they rebiopsied, they found the T790M. So the blood test was able to detect this before the tissue biopsy was, or we don't really know, but could have been that the tissue biopsy missed. So important aspect of why you want to do the blood testing along with the tissue biopsy in order to qualify patients for these therapies that truly can save or extend their life. So now, if you know the oncology market, you know it's a very large market with a lot of oncologists in America, and it's seen a lot of consolidation. And today, we're focused on various aspects to the business. We started out with the KOLs, and I think that's one thing we did well. I hired a sales team that had experience and a lot of relationships already, so we got in front of key opinion leaders with our testing so they would at least know about us. That's important because if a community oncologist wants to call up their mentor at the teaching hospital, we want them to know about Biocept. So even if they don't use us, and a lot do, they'll at least hopefully have a good impression of our company and the testing we offer. From there, though, we're starting now to market more and more kind of the tier 2 and tier 3 where the patients are treated. So most of the treatment in America is actually given in the community and will be influenced, though, by those tier 1 medical centers. So today we have salespeople where you see the red stars all around the country. We've got 11 sales folks out today and wanting to grow that as we go throughout 2017. We market to medical oncologists. Medical oncologists are the physicians that actually give the chemotherapy and the treatments. And since this is blood, they can draw the blood right in their office and send to us. So it's an efficient way that we're able to get the information straight to those who need it the most, the medical oncologists, so they can treat their patients. In addition, we also work with pathologists and surgical oncologists at times. Most of the volume that comes through our door comes from medical oncology. We also partner with pharma, and as I said, we've got 3 pharma projects right now that we're working on as a company. Very experienced sales team with greater than 5 years experience. We also have an internal staff to support them in customer service, in marketing, and administration. Now you can see they're doing a great job. We've really grown over the past year, and you can see that last year we had about 26% compounded quarterly sequential growth, quarter over quarter. And when you look at this slide, you can see that the blue is billable cases. These are cases we get from physicians, and they use that to make treatment decisions, give us the billing information for that patient. The reddish color is what we get from pharma, and the green are the studies we're involved in. We don't get paid, but those are volume coming through our lab. Now, one of the things the health plans like about us is the fact that we're focused on these biomarkers that matter. So therefore, an oncologist can order just what's most needed for that patient without having to order an expensive large panel like some of the other folks in liquid biopsy only offer. So that's led to a lot of contracts. And in fact, as I said, we just announced another agreement with Blue Cross of Texas earlier. And you can see that's driving our revenue growth. As we've gotten our billing processes worked out, our contracting worked out, that's paying off in the revenues that we're gaining as a company. And you can see we're growing quarter over quarter there. Just had our first $1 million quarter in our last publicly reported quarter back in Q3 of 2016. So we'll be reporting our Q4 results probably in the second week of March, somewhere in that time frame. So, you know, as I said today, we're a central lab. The test is all done here in our CLIA lab right here in San Diego. And we want to eventually go through the FDA and be able to market our testing so it can be done in hospitals all around the world. But today we can do testing in our own lab under what's called LDT. We do it as an LDT, lab developed test, and that's commonplace in the industry. Much of the laboratory and specialty testing that's done in America today is done through that LDT format. We do, though, want to do a new aspect to it with a partnership model right here in stage 2, which we're planning to launch this year. So that's where we would be able to allow the hospitals to share in some of the revenue with the testing we provide in 2017 as we move more and more towards the kit strategy in 2019. So milestones to look for in our company is continued commercial growth. You know, we'll enter into more and more partnerships, sign more health plan agreements. We'll have more and more data coming out. Remember, we're involved in 24 different studies today. Launch additional markers, which drives our growth. And then we'll also be launching a study that helps to value the capture of the cells, that helps to make our kit strategy in the future a reality. So folks, thanks a lot for listening to our presentation today about Biocept. I'm happy now to answer questions about the information that I presented.
Thank you, Mr. Nall, for that informative presentation. Now, before we get started on the question and answer session, I just want to remind our audience how to submit questions. You can submit the questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many questions as we can, and if not, Mr. Nall will be able to answer those via email post-presentation. Okay, now our first question is, Mr. Nall, are the Biocept target selector offerings used for cancer screening or monitoring?
Well, thank you, Christina. That's a good question. So as we talked about earlier, you know, there's different aspects to liquid biopsy today. And today, Biocept's focused on profiling primarily, which is where we're able to help where a tissue biopsy is either inadequate or impossible to get. So in other words, there's not enough tissue or the patient's just too sick to even have a biopsy. So we're able to get that information to an oncologist that they need to find out how to treat that patient. Next, we're doing monitoring. So that was one of the things that was asked, is about monitoring. And so that's an area that's really growing for us because physicians need to look to see, is the patient responding to the therapy or not? And with a molecular test, a blood test that we can do, we can start to monitor that patient every time that the oncologist sees him. So we're really in profiling and monitoring primarily. Screening will be something we're interested in the future, but there's a lot more data that the whole industry has to gather to find out what this would mean if we can find material in your blood that would indicate you may or may not have cancer. So a lot more work to do for a screening assay in blood today, but today we're focused on profiling and monitoring primarily.
Okay, next question. What is the strategy around FDA clearance of the assays, or is it even needed?
Oh, that's a good question as well, because I know it's a confusing topic. If anybody actually knows about our area of laboratory medicine, there's been a lot of talk over the past few years about this idea of lab-developed test, LDTs. If you Google it, you're going to get a lot of information. But today, you know, most of the laboratory testing that's done at the molecular level is done as LDTs in America. And some of the most well-known assays, such as Oncotype DX or the Foundation One, are LDTs. So the FDA does not currently regulate that part of our business. That doesn't mean that there aren't important validations that are done. In order to offer a test in America, you need to do an analytic validation that we described earlier, and then most high-quality labs will go ahead and do clinical validations as well. So you have both those aspects to be able to meet the threshold to offer a test for patient care. In the future, though, we want to go through the FDA to be able to then take the testing we do in our own lab and market that to hospitals. So that's the difference. I can actually do the testing in my lab as an LDT today under CLIA and the CAP accreditation that we have. But then for us to take the tests we do, make it into kits, and then be able to market it for specific use, we'll need to go through the FDA, and that's something we want to do in the future.
Thanks, Mr. Nall. We have some great questions coming in, but we're almost out of time. Let's fit one more in here. How about this one? Why is it important to detect and analyze both cells and DNA fragments?
Oh, I love that question because that's one of the key differentiators of Biocept. The ability to be able to look at the intact cells, called circulating tumor cells, as well as cell-free circulating tumor DNA, all out of that same standard blood sample. So that's important because there is information you can only get out of the cell that you simply can't get out of cell-free DNA. So it's just biologically impossible. So we can do protein expression, we can do DNA amplifications. These are just different tests that you need to get a result on so an oncologist knows how to treat their patient. So when we go see a medical oncologist, very often they don't necessarily care that we can do CTCs and cell-free DNA. What they love is that they can get a protein marker such as ER for breast cancer and HER2 for breast cancer. At the same time, they can get a mutation as well. So in lung cancer, they can get PD-L1, and they can also get ALK and ROS1 and EGFR mutation. So in other words, we can offer a more comprehensive menu because we can get tests that can only be done in the cells and cell-free DNA out of that same blood draw. That's very important for their treatment planning, and that allows us then to be used as a true surrogate for that tissue biopsy.
Thank you. And as a reminder to our audience, if we didn't get a chance to get to your question, it will be answered via email afterwards. Mr. Nall, do you have any final comments for our audience?
Well, thank you all for the time together today. I enjoyed sharing our story of Biocept, where we're delivering personalized medicine from a liquid biopsy. You know, just a couple things to remember. We're doing our testing today in San Diego in a CLIA lab, and we're able to help thousands of patients this way. More and more, we are getting paid for the work we're doing, which is an important thing. But the most important thing we do every day is be able to qualify more patients for these targeted therapies that truly do extend and save their life. So that is the most important thing. Thank you very much.
A big thank you again to Mr. Michael Nall. I would also like to thank our sponsor, LabRoots, for making today's educational webcast possible. Now, today's webcast will be available for on-demand viewing through May 23rd, 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks so much for joining us, and we hope to see you next time. Goodbye.